My research focuses on understanding how the cells in our body normally prevent us from getting Parkinson's disease. Parkinson's disease, at its core, is a movement disorder. It's a neurodegenerative disorder. So it's our brain, over time, loses the ability to communicate messages. And if you see here in a healthy brain, normally there's a mostly simultaneous connection between the part of our brain that wants to move and the part of our brain that then tells the muscles to move. So if you're going to reach for a fork, you make the decision to reach for a fork, and then another part of your brain tells the muscles in your arm to start moving. In a Parkinson's patient, there's a disconnect between the part of your brain that wants to move and the part that tells the muscles to move. There isn't the ability to tell your body to move in the way that you want it to. That's why the main symptoms of Parkinson's disease is the inability to move, or in some cases, inadvertent movement, tremors, and losing the ability to walk properly. And while we've known about Parkinson's, it's been characterized very well medically for the last century or so, we've known very little about what's going on inside the body to lead to this disorder. And in the last couple of decades, we've understood better the genetic basis for certain kinds of Parkinson's disease. And that is that in Parkinson's disease, often there are mutations in genes that encode for particular proteins. Now, a protein is your cell's machine. It's kind of the factory robot. Your cell is the factory that gets everything done. Proteins allow you to see, they digest your food, they form your muscles. Proteins really do everything. And there are particular proteins in certain kinds of Parkinson's disease that aren't functioning properly. And I'm looking at one of those proteins. To get a better idea of what that protein is doing, we have to have a good idea of how your cell is built up and how the cell provides energy. So you see here we have an image in the top left of an individual, one person. And if we break down that one person, we're all made up of trillions and trillions of cells, about 35 trillion cells. And while they're diversified highly depending on where they are and what their function is, so for example, a cell in your eye is going to be very different from a cell in your heart, or a cell in your big toe, or a cell in your stomach. But at the core, they all have a similar structure and a similar way to generate energy. And if you remember from your school days in biology, you probably heard about the mitochondria or mitochondrion. These guys make up the energy network of your cell. They're the cellular powerhouses, and they provide the vast amount of energy that we need. And the reason I bring up the mitochondria is that we've identified the gene mutants in Parkinson's disease, the proteins that they encode, the molecular machines that are not functional in Parkinson's, are responsible for getting rid of bad parts of our mitochondrial network. Just like man-made power grids and power systems, certain generators and certain power plants will get old and fail and have to be repaired or even demolished over time. It's the same thing with your mitochondria. As you age, the network can get damaged, and certain parts of the mitochondrial network might need to be completely removed. If you don't remove it, you get a buildup of bad mitochondria that are gunking up the cell and are detrimental and can possibly lead to neurodegenerative disorders like Parkinson's disease. So one of the machines that I look at, Parkin, the Parkin protein, is a key protein we found out in removing very badly damaged mitochondria and targeting them for the cellular garbage disposal, you can think of, or the recycling plant. And so my goal is to get a better understanding of how Parkin identifies damaged mitochondria and how it helps remove them before that damage interferes with the larger network and before you can develop a neurodegenerative disease like Parkinson's. Now, as you can imagine, looking at what the Parkin protein is doing on the mitochondrial network is very challenging because you're trying to look at the insides of a human cell And if you look at your hand, well, it's made up of trillions of cells, you can make out maybe wrinkles, hairs, joints, but cells are tinier than what we can make out with the human eye. And we want to look at a machine inside the cell. It's like trying to look at a robot on an assembly line when you can't even see the factory, it's that tiny. So we use microscopes to do that. And as you can see here on the left and the right, we have images from two different kinds of microscopes, and we can correlate them together. Now on the left, you see a fluorescence microscope. And this kind of technology has been around almost for a half century now. And what we can do is we can take those little machines, the proteins. In my case, we take the Parkin protein, and we attach a tag on it that glows yellow. It's called a fluorescence tag. That's why this technique is called fluorescence microscopy. And because we can visualize that tag, we can then see where in cultured human cells the protein of interest is. So I can identify where my Parkin protein is. The problem with this technique is it's limited by the wavelength of light. What I mean by that is that light is actually, we can think of it as having a size. So we can't see anything smaller than the wavelength of light. So we're limited in that respect. And we're also limited in that the only thing that we can see is what's tagged. So while we can identify the tagged protein, Parkin, very well inside the cell, we don't see everything else. We don't see the nucleus, we don't see the endoplasmic reticulum, the mitochondria, we don't see all those things that you learned about in your biology classes that actually make up the cell. And so we combine this fluorescence microscopy technique with what's called electron microscopy. And electron microscopy works by shooting a beam of electrons through the sample. In this case, the same sample that's on the left is imaged on the right. And electron microscopy works just like the X-ray in your doctor's office works. Where in X-ray, you shoot X-rays through your body, and then you see what the X-rays can't pass through. You see your bones, your skeleton. Same thing for electron microscopy. What's more dense, in this case, what has density from proteins or from the lipids that make up the membranes, the bags around compartments in your cells, those show up all over the electron micrograph. So while we can't see specific tag things in electron microscopy, we can make out at a very high resolution the entirety of the cell. So if we combine the tagging of fluorescence microscopy to the high detail and the ability to see everything with electron microscopy, we can then localize where this Parkin protein that's mutated in Parkinson's disease is on a specific mitochondrion and get a better understanding of what it's doing to deal with the damage and to remove it. And so, as you can see here, I've identified in the fluorescence the particular protein of interest. In the bright yellow there, that's my Parkin protein. And you can see in both images in the top right, the nucleus, which is outlined, and the cell exterior in the bottom left for orientation. And you can also make out in the right, in the micrograph, the mitochondria, and I've identified those. And we can use these artificially added correlation beads that I've identified in the left image. These beads are special because I add them to my sample. They're not normally present in the cell, but they fluoresce in all different channels. They give out all different kinds of colors, so they're easy to identify from my signal in fluorescence. And unlike everything else in the sample, they're something that shows up in both fluorescence and electron microscopy. And since it's the same sample and they're in the same position, we can then use these correlation beads to map out where my fluorescence signal is on the electron micrograph and get very high precision correlation. And that's what you can see here. We've done the correlation. You can see the fluorescence overlaid now with the correlation beads, and we've done a software program that'll do this high precision correlation. And we can see the signal of interest from the Parkin protein on a particular mitochondrion. And now I can zoom in and take a high magnification electron microscope image of this particular mitochondrion that we know now has been targeted by Parkin. And we can understand why. We can start to understand by acquiring these data like this, is why Parkin has targeted these mitochondria, and why this one is sick, and what's being done to remove it. So we can already make out from this one areas of very high density, that's very black, inside the mitochondria. That's most likely aggregation of proteins that have clumped together and are stressing out the mitochondria, which might be one reason why the Parkin protein has targeted this mitochondrion for removal. So to back up and do an overview here, what we're trying to do with this correlative technique is piece together how our cells deal with bad mitochondria and normally prevent Parkinson's and other neurodegenerative diseases from developing, possibly from a buildup of these gunked up, dysfunctional mitochondria. And if we understand how our cells normally work and are able to prevent Parkinson's disease from developing, we can better hope to treat and possibly prevent Parkinson's from developing in cases where our cells are unable to cope with the damage.